we are learning human reproduction. In that, today we are going to discuss about the topics fertilization and implantation, pregnancy and embryonic development. Nandini?
Yes, ma'am.
Yeah, shall we start?
Yes, ma'am.
Yeah, okay. Yes, first, what is fertilization? What is the definition for fertilization, Nandini? Fusion of?
Male and female gamete, ma'am.
Yeah, where it is happening in human? Where it is happening in human?
Ampulla.
Yes, yeah, this part is ampulla. This part is ampulla, and here the fertilization is happening. So what is happening is during copulation, the male gametes, that is sperms, are introduced into the female reproductive system through vagina. Okay, so the sperms, they will enter through cervical region and through uterus, and they reach the ampulla where the egg is waiting. So how long the egg will be waiting? How long the egg will be waiting?
For
What
2
is
days.
the for 2 days? What is the viability of sperm?
3 days.
Yeah, 72 hours. And again, what about the sperm viability in epididymis?
4 weeks.
4 weeks. Yes, very good. So I think, yeah. So during copulation, insemination is happening. What do you mean by insemination? Releasing of semen into female reproductive system. Okay, so where in the female reproductive system the semen is released? In the vagina. Okay, so this sperm which is released into the vagina, it travels through, see here, sperms, it is released into the vagina, travels through cervical canal, uterus, isthmus, and reaches the ampullary region. And like that, when the ovulation will happen? When the ovulation will happen, Nandini?
At 14th day, ma'am.
Yeah, 14th day of menstrual cycle. So what do you mean by ovulation? Releasing of ovum from? Releasing of ovum from?
Graphene follicle.
Yeah, very good. So what is the status of ovum? Whether it is primary oocyte, secondary oocyte, oocyte. At ovulation stage, I'm asking.
Secondary oocyte.
Yeah, yes, very good. Secondary oocyte. Arrested at?
Metaphase 2.
Yeah, very good. Arrested at metaphase 2. So that is released, and it is captured by this finger-like projections called fimbriae, and the egg is traveling to the ampullary region, okay, through infundibulum. So this is very important. That is path of sperms in the female reproductive system and path of ovum. Okay, so this ovum which reaches this ampullary region, it will be waiting for 2 days. Only 2 days. Okay, so during these 2 days, if copulation is happening and insemination is happening, there is a possibility of fusion of gametes, like the sperm is coming from these areas and reaches this ampullary region. So when this egg is waiting for these 2 days, during that time copulation happens, means fusion of gametes is possible. Okay, and that too, if all other environment and hormone level, everything is fine, this fertilization will be possible. So each and every copulation does not lead to pregnancy. Whenever exactly, what is the exact copulatory period to get pregnant? Do you have idea? Exactly during the menstrual cycle, if copulation is happening between 10th to 17th day, 10th to 17th day, if copulation is happening, there is a possibility, that is the possibility for pregnancy will get increased. Okay.
Okay, ma'am.
Yeah. And again, here, what is happening after the fusion of gametes? After the fusion of gametes, what will be formed here in the ampullary region? Zygote will be formed. So during this fusion, so many reactions are happening during this fusion. That is acrosome. We saw the structure of sperm. What is the role of acrosome? It is having zonal license. Okay, license. What do you mean by license? This consists of enzymes. What are the enzymes present? Hyaluronidase. Hyaluronidase. What is the role of this hyaluronidase? This will digest some of the ground substances. Okay, and again, if you take second enzyme, that is corona penetrating enzyme. So this will penetrate, this helps to digest the corona radiata layer. Okay, and apart from that, zona license are present. That is zona pellucida layer is digested. Zona license help in the digestion of zona pellucida. Okay, so in this way, with the help of this acrosomal reaction. So what do you mean by acrosomal reaction? The acrosome, which is having all these license, will be released when it reaches the egg. So due to that, the sperm head enters into the ovum. So during this entry of sperm, this secondary oocyte will complete meiosis 2. Meiosis 2 is completed during fertilization. Questions will be asked. That is meiosis 2, when it is getting completed, during fertilization, before fertilization, after fertilization. Like that they will ask. So when the sperm head is entering, that is the signal for the secondary oocyte, which is arrested in the metaphase 2, it continues. Okay, prophase, metaphase, anaphase, telophase, and again it produces one more polar body and ootid. Okay, so then here both are haploid. Sperm and egg both are haploid. They fuse together to form the zygote. So yeah, see here exactly at 14th day, this is Graafian follicle and this is ovary. So from ovary, the egg is released as secondary oocyte, and it is waiting in the ampullary region, and sperm is traveling through the vagina, cervical canal, uterus, isthmus, and ampulla. Okay, when it reaches, this is called monospermy. So many sperms will be reaching this ampullary region, and in that only one sperm is entering into the egg. Okay, that is called as monospermy. Entry of one egg. Okay, so this fertilization requires 3 different reactions. One, as I explained, acrosomal reaction. Second is cortical reaction. So here what is happening in cortical reaction? The egg cortex region, which is having cortical granules, these also release chemicals, okay, which is helping in this fertilization. And the third reaction is fertilizin and anti-fertilizin reaction. Fertilizin and anti-fertilizin. Anti-fertilizin. So this fertilizin is produced by egg. Both are glycoproteins. Anti-fertilizin is produced by sperm. So what is this? What is the role of this? Means this attracts the gametes. Okay, so these are very much helpful for the attraction and fusion, because one is having negatively charged amino acids. Both are glycoproteins in nature. Just you imagine these proteins which are having positively and negatively charged proteins, they will attract each other. So that is also one of the reasons for this perfect fertilization process. So these 3 reactions together makes fertilization completed. Okay, so after this zygote will be formed. And that zygote, this is the nucleus of egg and nucleus of sperm fused together to form single, that is all the chromosomes of sperms and eggs fused together. Okay, so day 1 single celled zygote will be there, and first cleavage will happen. What do you mean by cleavage? Again, during the development of embryo, there are different stages. So from one cell, we are going to develop entire organism. So that is really a miracle. And again, first single cell, from here, this undergoes mitosis. This undergoes mitosis. So this single cell, it is converted into 2 cell to 4 cell, 4 cell to 8 cell to 16 cell, 16 cell to 32 cells. Okay. So here 16 cell containing embryo is called as blastula. Okay. That is blastula. And again here the morula. Morula means that is here the cells which are formed from the zygote, they are called as blastomeres. Blastomeres. Okay. So here the size of the cells are getting decreased when compared to the zygote. And again, these blastomeres, if they are forming 8, they are called as morula. So from zygote, we are forming first morula by cleavage process. And from morula, blastocyst is formed. So this is called as blastulation. So here this blastocyst, again it is divided into 2 different parts. That is, outer layer is called as trophoblast, inner layer is called as inner cell mass. Okay, so see here, after that the implantation is happening. After blastocyst stage, that is helpful for implantation. What do you mean by implantation? Because from the zygote to blastocyst formation, the nutrients needed for these cells are provided by endometrial lining. Please remember, this question will be asked. So endometrium of uterus is secreting the nutrients which are needed for the conversion of zygote to blastocyst. After that, more nutrients are needed. Okay. So until the development of placenta and umbilical cord, okay, this uterine wall is helping this embryo to develop. For that, implantation is needed. Okay. So after blastocyst formation, implantation is happening. So day wise, it is given clearly in this picture. Please go through that. From day 1 to day 5, blastocyst is formed. So it will take around 8 to 9 days, at least 1 week, not less than 1 week. This implantation is happening. Okay. So after that, this blastocyst is converted into gastrula. So I will explain what is gastrula. And again, this process of conversion of blastocyst into gastrula is called as gastrulation. Okay, is it clear, dear Nandini?
Yes, ma'am.
Yeah, okay. So from zygote to embryo, all the divisions are only mitosis. So I think it is clear what are blastomeres, what is morula, what is blastocyst, what is gastrula. Okay, yeah. See here. See, zygote by cleavage process, it is getting converted into morula. And again, morula is converted into blastula. Specifically, we have to call this as blastulation. So from blastula, after that, the outermost layer is called as trophoblast. And the inner cell are called as inner cell mass. See here, inner cell mass. The outer layer is differentiated into trophoblast. So from this inner cell mass, germ layers are formed. That is called as gastrulation. See, gastrulation. What do you mean by gastrulation? Gastrulation is germ cell layers formation, like endoderm, ectoderm, mesoderm. Endo, ecto, and mesoderms, from where they are formed? From inner cell mass. Again, question will be asked. Then what is the role of this trophoblast? So this trophoblast layer gives nourishment, and it is very much helpful to drain nutrients from the uterine wall, one thing. And it is forming, this trophoblast is forming the placenta. Okay, first it will form chorionic villi. So all the membranes like placenta, chorion, amnion, allantois. So these three are formed from the trophoblast. Ectoderm, endoderm, and mesoderms are formed from the inner cell mass. Okay, so from this ectoderm, endoderm, and mesoderm, all the organs and organ systems are developed from all these three. Got it? So this three-layered structure is called as gastrula. Clear?
Yes, ma'am.
Yeah, see here, let us read. That is, fertilization happens only if ovum and sperms are transported simultaneously. So it is not necessarily that all the copulations lead to fertilization and pregnancy. Whenever exact that 2 days when the egg is waiting, if the sperm is meeting the egg, fertilization is possible. So how the sperm is getting entered into the egg? By three different reactions. Acrosomal, both are functioning in that process. That is, acrosomal reaction is produced by sperm, and cortical reactions are produced by egg. Fertilization and anti-fertilization reactions are produced by both sperm and egg. Okay, and again, this sperm contacts the zona pellucida of egg. Okay, question will be asked. There are so many membranes, and again here the sperm can contact zona pellucida, and it is producing all the enzymes to digest all the layers which is present as outer covering of egg. Okay, so it induces the membrane that block the entry of additional sperm. So only one sperm that will be allowed to enter the egg. Okay, and again, the secretions of acrosomal reactions help sperm to enter the egg cytoplasm via zona pellucida and plasma membrane. So this zona pellucida and plasma membrane are digested by acrosomal reaction. So this causes second meiotic division of the secondary oocyte to form oocyte or ovum and the secondary polar body. Okay, so the haploid nuclei of sperm and ovum fuse together to form diploid zygote. So how many chromosomes will be there in zygote? 46 chromosomes. So in this page, if you have any doubt, please ask there.
No, ma'am.
Yeah, okay. Yes, see here, this already I have explained, and what type of cleavage is followed in human embryonic development. That is very much important. So here, unequal, holoblastic, indeterminate cleavage. So please remember this also. So what type of cleavage, unequal cleavage is happening because the cytoplasm is getting reduced in blastomeres. Okay. So again, holoblastic means the cell is dividing as a whole like this. So that is holoblastic and indeterminate cleavage. Okay. And again, that is the embryo with 18 to 16 blastomeres are called morula. Morula continues to divide to form blastocyst. Cleavage in man is holoblastic and indeterminate type. Clear?
Yes, ma'am.
Yes. Yeah, that's all. See again here, after attachment, that is implantation. After attachment, uterine cell divides rapidly and cover the blastocyst. So you just imagine this is the blastocyst and the outer layer is called the trophoblast. So this trophoblast is forming chorionic villi, and then it is developing into placenta. So this placenta is connected to fetus by umbilical cord. So until this formation of placenta and umbilical cord, this trophoblast drains nutrients from uterine wall. Okay, so before the formation of trophoblast, the endometrial uterine endometrial wall will be secreting the necessary substances needed for the conversion of zygote to blastocyst, and this inner cell mass again it is developing into organs, different organs. Okay, so that is the this blastocyst which is getting embedded into the endometrium wall is called as implantation. Okay, And this I have explained what is cortical reaction, what is fertilization and anti-fertilization reaction, and apart from that, acrosomal reaction. Acrosomal reaction is also needed for fertilization. So what is acrosomal reaction? That is lysins are produced. Yes. So I think I made it clear. If you have any doubt, you can ask.
No, ma'am.
Yeah, okay. Shall we move on to pregnancy and embryonic development?
Yes, ma'am.
Yeah, okay. So after implantation, finger-like projections are called. See here, this is trophoblast layer. This is trophoblast layer, and again, this is called finger-like projections are developed from the trophoblast layer, and again, it is converted into placenta. So this is placenta, and here the fetus is there. This fetus is getting, after some days, it is getting nutrients from the mother's blood directly, all the nutrients, oxygen, everything. And again, this placenta is very much acting as a mediator for transporting the nutrients and waste materials from mother to fetus, fetus to mother. And this placenta is connected to the fetus by umbilical cord. See, this is umbilical cord, which is directly supplying the nutrients which are received by the placenta. This is like, you consider this as diffusion membrane. Placenta has diffusion membrane. Okay, so see, first after implantation, that is after the egg, after the blastose is embedded in the endometrial wall, finger-like projections started from the developed from the trophoblast layer. Okay, and again they are surrounded by uterine tissue and maternal blood. So this chorionic villi and uterine tissue both together will form the placenta. Again, question may be asked. So placenta is developed by trophoblast and uterine tissue also. So both together forms the placenta. So again, what is the major role of this placenta? Means it is the functional unit between the mother and fetus. Okay, so again, you can just imagine like diffusion of all nutrients and diffusion of nutrients and waste materials. Diffusion of nutrients and waste materials are happening through that is transported through the placenta. Okay, and again, placenta is connected to the fetus by umbilical cord. Yeah, you just imagine. See, this is like villi-like substance which can absorb and transport to the umbilical cord, and umbilical cord is transporting the nutrients to the fetus. Okay, and development takes place. Yeah, see, other than this, other than supplying nutrients and removing waste material, other than that, what are the other functions of placenta? Let us see. So that is, it is acting as a barrier between fetus and mother by supplying all the nutrients, oxygen, and eliminating carbon dioxide and other excretory waste materials. And one more function, it is acting as endocrine gland. See, question how they will ask, what are the hormones especially exclusively produced during pregnancy? Okay, so these are the hormones you have to remember: human chorionic gonadotropin and human placental lactogen, and estrogens, progesterone, and relaxin are the hormones which are produced by placenta. So little amount of estrogen and little amount of progesterone is produced by placenta also. So apart from that, large amount of progesterone is produced from, tell me.
Corpus luteum.
Yeah, very good. So even though large amount of progesterone is produced by corpus luteum, this placenta is also producing progesterone. And what is the role of this progesterone? Tell me.
Maintain pregnancy.
Yeah, maintains pregnancy and maintains endometrium also. Maintains endometrium also. So if that 2 days, egg will be waiting. There is no copulation is happening. There is no fertilization is happening, means automatically the egg will be degraded. And again, that a corpus luteum which is waiting for this event to happen, this fertilization event to happen. If it is not happening, that will be converted into corpus albicans. So when it becomes corpus albicans, it does not produce progesterone. Progesterone is needed for maintenance of endometrium. So if the progesterone level reduces due to the failure of fertilization, okay, so during that time menstrual flow will happen. Okay, and what is the role of this relaxin? And this relaxin is also produced by ovary. That is, corpus luteum also produces this relaxin also. What is the role of this relaxin? No idea? Okay, so during childbirth that cervical region, it should get relaxed for the to create that what to say, the ejection reflex. Okay, so for that this relaxin hormone is needed during parturition. Clear?
Yes, ma'am.
Yeah, how we will identify pregnancy? Have you ever seen the testing of pregnancy? Even in many movies they might have shown.
No, ma'am.
You don't know?
No,
See here, just a plastic strip will be there. Okay, in that one well will be there, one small well-like structure will be given. Okay, see here, this human chorionic gonadotrophin, this will be released soon after the formation of placenta. So this HCG is produced only by placenta. So placenta, when we will be forming, when the female body will have this placenta only after pregnancy. Okay, so this HCG will be present in urine. Okay, so this HCG is present in urine of pregnant women. Okay, so in this strip already we have coated with anti-HCG, human chorionic gonadotrophin. So if we add urine which is having HCG, urine of pregnant women. So by collecting the urine itself, we can identify whether the women are pregnant or not. So if this urine is having HCG, this anti-HCG will react and form bands. So here already the both anti and HCG are loaded in one band, and here only anti-HCG plus HCG will be loaded in one place, and in another place here only anti-HCG will be present. So if the urine is having HCG, we will get two lines. Understanding, dear? Understanding? Yeah, see here. So this is like a strip, and here that is we cannot see the lines, it will be plain, and here this is called as standard. Okay, so this is having both HCG and anti-HCG. So if I'm adding urine without HCG, no HCG, so if I'm not pregnant, my urine I'm collecting and I'm pouring, means my urine there is no HCG is present. Okay, so what will happen means there is a like it is like a paper like thing. You just imagine like that inside that plastic vial, and again that will move. This urine gets absorbed and reach this area. So already see here, when it is getting wetted, this HCG and anti-HCG will produce this color will be produced. But if HCG is there, again in the next place, here there is no HCG, only in this area only anti-HCG is present. Okay, so if the urine, if it is having HCG, that will produce color like this. So in the presence of pregnancy, we'll be getting two lines we'll be getting. And in the absence of pregnancy, you will get only one line. Are you getting here?
Yes, ma'am.
Yeah, that's all. So by this way, we can identify the pregnancy. Okay. Yeah. So again, what are the hormones which are produced exclusively during pregnancy? Means all these hormones, HCG, HPL, progesterone, estrogens, and relaxant. Okay. So apart from that, other hormones also raised during pregnancy. What are they? Cortisol, prolactin, thyroxine. So why this is needed? You know very well that the functions of thyroxine already we have discussed. That is, it is needed for metabolism. So during embryonic development, so much of metabolic reactions are needed to develop develop one more organism, new organism. So for that, you just imagine how much of nutrients are needed, how much of energy will be needed. So for that, this thyroxine is inducing, and even all the nutrients for the development of fetus, it is obtained from mother. So mother's metabolism will also get increased. Mother's requirements of nutrients will also be increased. Okay, calcium, folic acid, iron, because blood and other all cell division itself needs so many nutrients. Okay, even from one cell, millions of cells we are producing. So for that, so much of nutrients are needed. For that, metabolism should be increased in the maternal part also. Okay, so for that, and again prolactin, you know, that is prolactin will be released after that is during the stage of parturition. So what is the role of prolactin, dear? From where it is released? Prolactin, from where it is released?
Milk production hormone.
Yeah, yes. From where it is released?
Pituitary.
Anterior pituitary. Clear? Yeah. Yes, okay. So all these hormones together supports the fetal growth and even metabolic changes in the mother and maintain the pregnancy. So all these hormone levels are very much important, even though if the fertilization occurs, it should grow well till 9th month of the pregnancy. That is very much important, and that is maintained by all of these hormones. Okay, yeah. And again, see all the layers already we have discussed in structural organization itself. And again, that is endoderm, ecto, and mesoderms. So how they are getting developed into other organs. Uh, that is month-wise, they produce different organs. Okay, so from these three layers, all the organs and tissues are getting developed. For example, ectoderm is responsible for production of brain and central nervous system, everything. And mesoderm, if you take muscle, you can say. So like that, endoderm, all other organs, important organs, you can say. Okay, yeah. Yes, see here, that is how long this gestation period is happening. Okay, 9 months in human beings, and for cats, if you take totally 67 days. Okay, and dogs, if you take 64 days, almost 2 months, and elephants, if you take 22 months. So the organism which is having the longer gestation period is elephant, 22 months. Yeah. So what is happening during the embryonic development? So from zygote, after 30 days, we are forming the heart. Okay, that is the embryo is getting developing, and during at the end of first month, the heart is formed. Okay, and after that, so each and every month it is given, like day 1 fertilization is happening, and day 2 two cell stage, day 3 16 cell morula is formed, day 4 blastocyst is formed, day 8 again implantation is happening, and day 12 complete entry of blastocyst into endoderm, that is gastrula formation is happening during 12th day. Okay, and after that, if you take 28th day, heart is completely developed and it starts beating also. Okay, and after 7 weeks, jaws. If you want in detail, I can give 7 weeks, jaws and fingers, external ears, and all other organs are started developing. After 2 months, if you take, that is second month, limbs digits are developed, and at the end of the third week, which is called as first trimester, totally in that 9 months, we can divide that 9 months into three: first, second, and third trimester. Okay, and again in the first trimester, at the end of first trimester, major organs are getting developed, well developed, genital organs, reproductive system, everything is getting developed. Okay, yeah. And at fifth month, first movement of the fetus and appearance of hair on the head. What is given in the NCERT alone, I have mentioned here. And again, if you take blood formation, bone marrow, everything is developing here. Bone marrow formation also developing here. Okay, and again, end of second trimester. End of second trimester, body is getting covered with fine hair and eyelids separate, and eyelashes also formed. And end of 9 months, end of 9th month, ready for pasteurization. Okay, so you just remember what is the content given in NCERT, that is enough. Here you can expect matching type question. So question, matching type questions you can expect from this slide. Okay, after 1 month, heart is formed. Second month, end, limbs and digits are developed, and end of first trimester, all the major organs and reproductive systems are getting well developed. And fifth month, again, first movement of the baby is fifth month, and again appearance of hair on head. And second trimester, body, hair, eyelids, and eyelashes also formed. That's all. So if you have any doubt in this, you can ask me. And what are amniotes and anamniotes here? What is the difference between amniotes and anamniotes? This we discussed in animal kingdom. Do you remember, Nandini?
No, ma'am.
Okay. Amniotes and amniotes. See here, again, we discussed about the differences between viviparous, oviparous, ovoviviparous. Do you remember?
Yes, ma'am.
Yeah, what is the difference, dear? Viviparous means they will get, that is, the fetus will be developing with the help of nutrients which are transferred from the maternal blood to the fetus through placenta. Okay, that is called as viviparous. And oviparous and ovoviviparous means oviparous, fertilized or unfertilized eggs will be released out of the body, so that it is clear that there is no connection between the mother's blood and the zygote. So automatically, whatever the nutrients present inside the egg will be used when it is laid outside. For example, frogs, it is purely external fertilization. But if you take chick, they are again, they are laying fertilized egg. They are laying fertilized egg. And again, that zygote which is developing into a chick, that utilizes only the nutrients which are present in the egg yolk. Okay, but ovoviviparous, if you take, there is no formation of placenta. It is present inside the body only, inside the female body, but there is no formation of placenta. Okay, and like that here, amniotes and anamniotes. Here again, chorion, amnion, that is extra. Yeah, allantois. Those are not present in anamniotes. And amniotes, they are having presence of all these layers like amnion, chorion. What is the role of allantois? So these three are present in anamniotes, and that is absent in anamniotes. Sorry, amniotes, they are having these three layers, anamniotes, they are not having. And here, what is the role of amnion? Tell me. What is the role of this amnion?
Protection, ma'am.
Yeah, protection. Very good. And again, it is filled with amniotic fluid. Amniotic fluid. What is the role of allantois? Allantois, again, it is a sac-like structure which will collect all the waste materials. Okay, that is the role of allantois. Chorion is developing into placenta. Got it?
Yes, ma'am.
Yeah. Yes. So again, what are all the things you have to remember here? What is cleavage? What is bastulation? What is gastrulation? And again, already we have discussed about what are all the organs developed from endoderm, ectoderm, and mesoderm. You please make a list of those and please remember that also. And again, at 8 cell stage, it is called as morula, and 16 cell stage, blastocyst. And after that, 32 cells, they involve in again implantation. After that, what will happen? Trophoblast is happening, inner cell mass is happening. What is the role of trophoblast? All these layers, amnion, chorion, and allantois are developed from the trophoblast. Inner cell mass is responsible for gastrula formation. Gastrula means 3 layer, germ layer formation is called as gastrulation. Okay, and how many days the egg will be waiting, everything you have to remember.
Ma'am, gastrulation மட்டும் மறுபடியும் சொல்லுங்க ma'am.
இப்போ இங்க என்ன ஆகுதுன்னா zygote is there. Zygote is 1 cell. Okay, so அது 2 cell ஆகி 4 cell ஆகி 1 to 2, 2 to 4. 4 cell will be converted into 8, 8 into 16. Okay, so up to this is called as, like you can say, this is morula. This is morula, and again this is called as cleavage. And next is conversion of morula to blastula. That is called as blastulation. So gastrulation means conversion of blastula into outer trophoblast, and inner cell mass. So this conversion, again, this inner cell mass is developing into endo, ecto, and meso. Okay, so here this differentiation of these 3 layers in inner cell mass, that is called as gastrulation. Got it?
Yes, ma'am.
So in blastula, there is no differentiation of endoderm, ectoderm, and mesoderm. So for that, it is given clearly in 3 different colors. So this light blue is trophoblast, this dark blue is ectoderm, and this yellow is mesoderm, and this is endoderm. Got it? So the differentiation of inner cell mass into 3 different germ layers is called as gastrulation. Okay?
Yes, ma'am.
Yeah. So everything I made it clear, I think. So here it is very important. And again, when the meiosis 2 is getting completed is also very much important. And again, that is the ovum which is released, what state it is, and how many polar bodies are produced. Tell me how many polar bodies are produced.
When, ma'am? Whole 40.
Yeah.
Totally 2 polar bodies.
Yeah, very good. Very good. If in the options, if it is given like 2 to 3, you have to select 2 to 3 polar bodies, because the fate of these polar bodies is unknown. Whether it is getting divided or not, it is uncertain. Okay, so 2 to 3, if it is given, you select that. Okay.
Okay, ma'am.
That's all we have completed. Tomorrow we are going to complete this chapter and question discussion.